In general, as you know, pigs have a lot of similarities with humans, not only in terms of anatomy, being monogastric, but also physiology and genetics. They're three times closer to humans than, say, mouse models in terms of a lot of those studies. In terms of pathology, a lot of common diseases, and they make really good model.
Welcome, everybody, to the Swine Canada podcast. I am Dan Columbus, and I'll be your host for today's episode. And with me today, I have Dr. Ali Honaramooz, who is a professor of Veterinary Biomedical Sciences at the University of Saskatchewan. Welcome to the show.
Thank you very much, and thanks for the invitation.
Yeah. So because some of our listeners may not be familiar with you, you're not generally in the area that we have on the podcast, I'll just ask you to kind of give an introduction of yourself and your journey so far before we get to the topic today.
For sure. So my name is, as you said, Dr. Ali Honaramooz. I'm a veterinarian by training. I'm from Iran. I did my DVM in Iran. Then I taught as a university instructor for a few years, then decided to continue my education. So I came to actually Saskatoon, University of Saskatchewan. That was a while ago, and I did a PhD in reproductive biology and technologies. And that took me to the next stage, which I did postdoc at the University of Pennsylvania. At the vet school there. And although the work was also reproduction in general, it was more geared towards germ cells and transgenic animals, especially farm animals. And from that point on, I switched most of my research in that direction. And after 4 or 5 years, I was recruited back at the University of Saskatchewan, came here exactly 20 years ago. And I've been teaching and doing research at the vet college here.
As we were talking before, you've been in the news quite a bit recently, and we'll talk about why for that topic, in the next episode. But for this one, I think it's related because you do use the pig as a model for a lot of your research, which has the human implications, correct? Right? So today we're going to talk about what you've been doing, specifically looking at infertility and kind of addressing that issue. So maybe let's just get into that. You can introduce what you've been working on.
In general, as you know, pigs have a lot of similarities with humans, not only in terms of anatomy, being monogastric, but also physiology and genetics. They're three times closer to humans than, say, mouse models in terms of a lot of those studies. In terms of pathology, a lot of common diseases, and they make really good model. On the other hand, a lot of issues with human fertility or infertility are a bit difficult to be done properly because mouse models are not a good model in a lot of ways. For example, mouse becomes mature, sexually mature, would have their first sperm within four weeks after birth, which is quite early. And if you are doing anything to try to see if the development of those sperm production processes are affected, it's a whole lot different than, say, in humans. Pigs are a happy medium in that it takes them 6 months or so, 5-6 months, most breeds, to have the first sperm, and you have a long period of time to do any of those research related on factors that affect infertility. So we became interested in stem cells. More specifically, in the testis, we have a population of stem cells called spermatogonial stem cells because they eventually lead to sperm production. And just like any other stem cells, they have two main characteristics. They can renew themselves, so they're never depleted. They can perpetually live. And on the other hand, they can give rise to daughter cells that can differentiate and make other cells. In this case, spermatogonial stem cells sit in the seminiferous tubules in the testis, and forever they can renew themselves, and then differentiated germ cells become sperm at one point. So that gives them another advantage over all adult stem cells in that if you make a genetic change in those spermatogonial stem cells, forever that spermatogonial stem cell is going to produce transgenic sperm for the rest of that animal's life. So in a way, you have changed the progeny by changing genes in those population of stem cells. So that is very intriguing to us, and we did some original experiments in 2002. We published, we call those germ cell transplantation in pigs. You can look them up under my name. So in as a proof of principle, for example, we got testis tissue from the same pig in the morning. We took one testis out. In the lab, we did tissue digestion. We got cells out, testis cells, including spermatogonial stem cells out, and went back and injected them into the testis of the same animal. And we made some modifications in terms of dyeing those cells so that we can detect them fluorescent-wise later. And we noticed that they stick around for as long as we were sampling them. Then we got encouraged. We got testis from one animal injected into the other. And to our surprise, they weren't rejected, and they were there a while ago, for a while. So then because the markers that we were using had temporary staining on them, the fluorescent dye would wash out after, and we were really interested to see what happens if we leave them longer. In that case, we used an animal that had been genetically modified. It had a human gene in it, and we used those as donors, and we injected them into recipient animals. And we kept them for several years. And we kept on getting their sperm checked. And even up to 5 years after, we had those sperm that were transgenic. They had the original donor that was not related. The recipient animals were not immunosuppressed. And that was not expected, because we would expect the cells to be rejected, even allografts. You have to use either genetically very similar, like twins or inbred animals, or immunosuppressed animals, but these were not either. So that by itself opened a line of work that they have in human reproduction treatments. They call it germ cell transplantation. And the idea is that, say, a kid has a cancer, and unfortunately, 1 out of every about 700 kids would get cancer before the age of maturity. So if you're mature and you have cancer and you have to go under cancer treatments, be it chemotherapy or irradiation, there is a risk, a rather high risk, of becoming infertile, because those treatments can damage those existing stem cells in the testis. And infertility can occur in 10 to 50% of people who recover from that cancer. So with the advances in cancer therapy, thankfully 80+% of kids who get treatment would survive their cancer, but without any intervention, up to half of them would become subfertile, problems with fertility, or completely infertile. So, so the idea is that those kids, along with other preparations ahead of chemotherapy, for example, 1 week before those treatments begin, they take them to hospital. They take like bone marrow samples in case they have anemia after. The suggestion is to take small biopsies of their testis tissue from these, immature boys, and freeze them for future in case they do indeed become infertile and they want to do something to basically restore their natural potential for parenthood. And at the time when we did this, there was no protocol to freeze testis tissue, so we developed some protocols, and we ourselves didn't do much with human testis per se, being at the vet school and everything, but we had colleagues who took our proof of principle research and took it, and they're continuing thus far. And today, they are close to doing it clinically, and the idea is to maintain those in tissue biopsies in liquid nitrogen. Decades later, if they need it, they can go back, retrieve those samples, and do something with it. But again, those biopsies being very small, it may not be enough to repopulate the entire testis tissue after recovery from cancer. So the research also includes ways to expand those, either culture them in vitro, in petri dishes, so that the numbers increase and then inject them, or do something else. And that'll take me to other areas of research that we have done ever since.
Well, I guess one of my questions is, and you mentioned that the human side of it has kind of taken this work, so have there been attempts to maybe answer some of those questions as to whether this would be effective in a testis that has in an individual that's been gone through chemotherapy or something similar to that? Because I assume your research was in a healthy pig that you then just re-injected into, right?
So that the initial work, yes, it was, but then again, we wanted to make it as close to reality as possible. So what we did was to also make pig models of kids who undergo chemotherapy or irradiation. So we had there are agents like a chemotherapy agent called busulfan, for example. It's very effective, but it also has great side effects. So we injected those to pigs at various stages, be it in utero when they were administered to pregnant sows, or after birth, or we did irradiation of their pelvic area, targeting the testis to make sure we get a good model working. And after doing various different protocols, we have a good model that replicates almost what happens without any remedies. They become infertile. Those piglets, male piglets, become infertile. And so they are a good model. So we went back and injected the testis that we had frozen thawed into them, and we showed that it can recover that fertilization potential back.
So it's there. I mean, so you have tested that they are fertile, like they can actually produce offspring with this re-injected.
Yes, and we have shown that the progeny carries the genetics of the donor cell, not so much the recipient at that time. It carries 100% of that progeny.
Pretty exciting stuff. I love this type of thing because it shows exactly what we can achieve by research in pigs, and that it does have more benefit than just producing a pig with good bacon.
Yes, absolutely.
So,
Absolutely.
the other thing you mentioned at the beginning, that this was kind of an unexpected response, right? Where you expected that being able to take something from one and put it into a recipient of somebody, and you don't get the rejection, but that's
No,
not what you're expecting. So what is it about this particular germ cell line that makes them not undergo that rejection?
so I guess the short answer is we don't know yet completely. If it works, we don't question it, I guess. But we know that testis cells, especially the somatic part of testis cells, including Sertoli cells, so in the testis we have both germ cells and somatic cells, especially somatic cells and Sertoli's in particular, they convey immunosuppression to the neighborhood in a way, because when an animal is born, um, it doesn't have complete spermatogenesis. The immune system that got into work during fetal period did a survey of all cells that were present at the time. And because fetus doesn't have sperm or haploid cells that have only half of the number of chromosomes, they weren't there. So the immune system said, well, everybody now is friend. Later on, when you have haploid cells, all of a sudden they look odd and the immune system wants to attack them. So the testis has this. So within the testis, in terms of its anatomy, we have these long tubules called seminiferous tubules, where, like in a boar testis, if you stretch them and put them together, it could be 5 kilometers long. And that's why you get millions and hundreds of millions of sperm ready to go at a time, because all the spermatogenesis is happening in these tubules. These tubules are immunoprotected. So blood vessels don't go in, and those Sertoli cells are sitting inside that tubule and protect germ cells within it. So not only they put their hands together, they form what we call testis, blood testis barrier. Don't let any immune cell to go in, but they also release immunosuppressive elements within that environment. As a prime example of that, when people do pancreatic cell transplantation as a therapy for diabetes, because that's the problem, right? Pancreatic cells are not there. If they put Sertoli cells of the testis with those pancreatic cells when they transplant them, they're tolerated a whole lot better, day and night difference, because they convey immunosuppression to those pancreatic cells and let them be accepted. So that probably is part of the fact that we didn't see any difference when we did from the same animal auto transplantation, or we did between animals allo transplantation.
When consumed through the feed acidifiers modulate the pH of the gastrointestinal tract with acids in nutrient digestibility. Research has shown that by including acidifiers in diet, you can increase average daily gain, increase average daily feed intake, improve feed conversion, reduce the pH of digesta. Vivo Vital from DSM Firmenich is an ultra-pure form of benzoic acid that, when used as a feed acidifier, has shown to improve post-weaning pig performance. Learn more at dsm.com/Vivo Vital.
Yeah, it makes sense, if you think about it, that because the sperm don't really match the host, right? So it would be very bad if it was attacking them all the time. So it's pretty incredible. So I guess the next question is, is there any other areas that you've been working on specifically when we're talking about testes, like maybe other broader areas, or like other toxins other than like the chemotherapy and the radiation?
Alongside those experiments, after we did the germ cell transplantation, at the same time, we did what we later called testis tissue xenografting, xeno meaning from one species to another. So instead of having cells injected into the testis, we had chunks of tissue, small biopsies from pigs that we put under the back skin of these mice that don't reject cells from others. They are immunodeficient. We call them nude mice because they're also hairless. They're pink little things. So we suture those testis biopsies under their skin. And to our surprise, again, these were from like day-old piglets. We started observing that they got enlarged as time went by. And the samples that came back about 6 months later, they had full-blown pig spermatogenesis. So in a way, to save the mice, we put like 8 little grafts on the back of these mice. So they were scurrying around with 8 little pig testicles on their back, which were producing pig sperm. So we tried that with also goats and so on, and it worked. And at that time, we sent it to Nature, which, as you know, is one of the most prestigious multidisciplinary journals, and it was accepted in Nature. And when it came out in 2022, we had tens of different newspapers and radio stations and so on, which were interested in the potential application of that. And at that time, people mostly were thinking of doing it for humans. And obviously, that is one thing that can be done, but not so much to produce human sperm, because you don't want to risk that sperm that has been developed in a mouse and put it back in a human, right? But you have all of a sudden a model that you can study. Say that you're worried about a certain molecule, a certain chemical in the environment that you think it affects development of sperm. You think that, say, bisphenol A, which we know was made into baby bottles, and that later turned out to be one of the factors that contributed to lower fertility in humans. You could test that in these mice that are carrying human testis tissue. So in a way, you can test on human testis without actual use of humans. Of course, you don't want to do that. So that was another thing that we did. Later on, we observed that testis tissues are amazing in restructuring themselves, forming the whole testis tissue all over again. We call it de novo testis formation. So in a nutshell, if you digest the testis tissue into its components, single cells, and then allow them to just congregate, again, we in a centrifuge tube, for example, we spin them down, and we just scoop that pellet at the bottom of the tube, put it under the back skin of these mice. They form the testis tissue. It's as if nothing had changed, which is amazing. It's as if you blow up the building into bricks and mortars, and you come back 2 weeks later, and everything has gone back to its complete normal, including vasculature, all the tubules with Sertoli cells all around, germ cells in the middle, producing testosterone and everything. So that was another model that we started looking, because it's very hard to study organogenesis. You cannot do experiments in, say, a fetus within the uterus of a mouse, because there's so much hormones around that any intervention would change the whole environment. But in this case, you're actually seeing organ formation in front of your eyes. And that is because at birth, the newborn would have to have a functioning heart, functioning kidneys and lungs to survive. But reproduction is probably intentionally, for good reasons, are immature. and held back until the animal is physically and mentally ready to take the responsibility of parenthood. So testis tissue, it's as if it remains immature. Even after birth, you take it out, you disperse the cells, they form the organ again, which is amazing. So that by itself is another model that we have used.
Very, very interesting stuff. We're coming to the end of this episode. For those who are listening, if you want to learn more, we are going to have a second episode talking about more of Dr. Ali's research. So please stay tuned for that one. Before we go, though, I will ask, a take-home message or kind of a final statement that you want people to get from today's episode.
So basically, we love working with pigs because of multiple reasons that I indicated. We have access to your wonderful Prairie Swine Center. Our students come there and collect the freshly castrated testes. So we have a source of fresh testis cells and tissue, and you can do unlimited number of work with them. So if anyone interested in that regard, that is a wonderful model for reproduction, for stem cell research, and as we're going to talk about in the next episode, even formation of organs, pig organs for use in humans.
Yeah. No, great. Looking forward to the next episode. So Like I said, everybody stay tuned and we'll save the 3 questions that we ask everybody to the next one. So if you want to find out book recommendations, then you have to listen to the next episode too. Right. So give somebody an enticement to come back and listen. So just thank you for this episode. It's been great and looking forward to the next conversation.
Thank you very much.